a most amazing news for our viewers. It is still one of the biggest mysteries in science. How does a human cell, too small to see with the naked eye, divide and reproduce to ultimately become a human body made up of more than 30 trillion cells? From the moment sperm fuses with an egg, human embryo development involves a string of complex and little understood processes. Much of what is known about embryo development comes from animals such as mice, rabbits, chickens, and frogs, with research on human embryos very tightly controlled and regulated in most countries. But animal studies can only tell researchers so much. What happens during human embryo development, particularly in the crucial first month, remains largely unknown. The drama is in the first month. The remaining 8 months of pregnancy are mainly loss and lots of growth, said Jacob Hanna, a professor of stem cell biology and embryology at the Weizmann Institute of Science in Israel. But first month is still largely a black box. Being able to peer into that black box would open up a world of biomedical possibilities, allowing scientists to demystify and previously obscured window of embryo development, ultimately leading to a better understanding to miscarriages, congenital birth defects, and the side effects of medication taken during pregnancy. And some researchers believe they have found a way to do it that bypasses the need for eggs or sperm. Heartening advances in stem cell labs around the world are making embryo-like structures, a group of cells that acts like an embryo but cannot grow into a fetus. Recent breakthroughs in the field, the culmination of years of painstaking lab work, have generated hope and some alarm, raising urgent questions about the ethical status of these models. To that extent, they should be treated like human embryos and whether they are open to misuse. The embryo-like structures are essentially clumps of cells grown in a lab, which are similar than a grain of rice and represent the very early stages of human development. Before any organs have formed, they don't have a beating heart or a brain. The most advanced models revealed in September by an Israeli team that Hannah was part of show all the cell types that are essential for an embryo's development: the placenta, yolk sac, chorionic sac, outer membranes, and other tissues that an embryo needs to develop. The structures were left to develop for 8 days, reaching a developmental stage equivalent to day 14 of a human embryo in the womb, an important moment when natural embryos acquire the internal structures that enable them to proceed to the next stage, developing the progenitors of body organs. Hannah said they were the most accurate models developed so far, and unlike those created by other teams, no genetic modification had been made to turn on the genes necessary to generate the different types of cells, only chemical nudges. It is not only you put the cells together and they are there, he said, but you see the architecture. You start also seeing very fine detail.